Whose is this?
Charles Springer, Jr. Active transmembrane water cycling high-resolution NMR detection of life.
That's good. Okay. Thank you very much, Michal. And I want to thank Gil and Hadassah. Oops, let me go back here for a minute. For the invitation. And I'm so honored to be here for this awesome birthday party. And I have slides of Gil's that I'm going to show at the banquet, so I'm not going to show them now. What I want to tell you about is some very exciting work by my colleagues, my former student and now colleague Jim Balshai at Harvard Medical School, and particularly a postdoc, Yaji Zhang, that Jim and I have supported on an NIH grant for the last several years. Now, we all know that water in tissue is very important, and it is used to regulate cell volume, but we're going to see there's much more to it. And we all know this because if you increase the osmotic concentration on the inside, then the cells will swell. If you decrease the osmotic concentration on the inside or increase it on the outside, the cells will shrink. And in fact, in the last 30 years, a great deal has been learned about exactly how water crosses these membranes, and this happens. But it's really important, I believe, to note that almost all of what has been learned in recent years has been done from experiments like this, where you set up an artificial osmotic gradient that the experimenter induces, and you watch a non-equilibrium water flux to either shrink the cells or swell the cells. But in fact, it's been known for a long time, longer than that, going way back to the 50s and even the 40s, that there is equilibrium exchange of water across the cell membrane. This is a review article, a monograph by House. This is not Dr. Gregory House. This is a British House, 1974, reviewing this really old literature, and some of it was extremely elegant, using labeled water to study the equilibrium exchange of water across cell membranes. In fact, the earliest experiments by Erik Zeuthen in Copenhagen started only a couple of years after the end of the Second World War. Very elegant experiments with heavy water and a tiny Cartesian diver to measure equilibrium exchange of water. And they used the term diffusional permeability coefficient because they assumed that it was simple diffusion across the cell membrane. And here's a list that House made of these results, and what you might notice is that erythrocytes are all up here at the top. This is the correct old version of making a table where it means the value has been multiplied by 10 to the fourth. And so these are like 50 or 60 times 10 to the minus fourth centimeters per second. And roughly, the erythrocytes are all an order of magnitude greater than the other cells that have been studied. These are the diffusional permeability coefficients. And these are all good numbers. We've actually checked a number of them. Other people have. They're really quite accurate. What I don't think anybody ever actually did, as far as I'm aware, of making a simple calculation of what was the significance of those permeability coefficients. And I'll show you the equation in a minute. All you need to know is the size of the cell, and you could have known that this permeability coefficient represented an equilibrium flux of water of 10 to the 14th water molecules per cell per second, going each way. 10 to the 14th water molecules per second per cell going each way. That's what these permeability coefficients meant, although I'm not aware that anybody ever actually published that. There have been a number of people who asked the question, why is it that the red cells, the erythrocytes, have an order of magnitude higher permeability coefficient? They're tiny cells. Here's a cross-section of an erythrocyte on a 10-micron scale bar. I'm going to tell you about yeast cells. Here are yeast cells. Other cells are much bigger. And people thought that the answer to this question of erythrocytes was found in the 80s and 90s when these aquaporin proteins that are specific water channels were discovered, and Peter Agre won the Nobel Prize for this, because, in fact, red cells are rich in aquaporins. But it turns out that really isn't the answer. This is a very busy slide, and I don't mean for you to look at. A lot of it is to remind me of what to say, but it's a picture of an erythrocyte and a cartoon of the metabolism, and it's to remind me to tell you that of the flux of 10 to the 14th water molecules per second per cell, it turns out from all kinds of experiments but from a number of people that only about 45% of that flux is going through aquaporin channels. And in fact, from studies of non-equilibrium water fluxes, those aquaporins are only using about 15% of their maximum capacity. So something else is going on to give you this 10 to the 14th water molecules per second per cell. And I'm going to give you a hint as to what that might be. Well, at the same time almost at the same time, NMR people, I know you can't read this slide, but NMR people have been studying equilibrium water exchange for many years. This is a review article from 1989, 20 years ago, really started with Conlon and Althred in 1972. So for 40 years, this is a list of erythrocytes, these are other cells. People have been studying equilibrium water exchange by putting a paramagnetic species on the outside and, in our modern notation, changing the shutter speed so that the exchange appears slow enough that you can measure. And among these MR scientists were some really outstanding MR scientists. Here's a paper by Hadassah Shinar and Gil Navon studying erythrocytes. And the general result of these studies show you that if we characterize equilibrium water exchange by the mean lifetime of water molecules inside the cell, tau I, they cover a huge dynamic range. For the human erythrocytes, many people, almost everybody, get a value of about 10 milliseconds for the average lifetime of water inside the cell. On the other hand, as far as I know, the record large value, it was determined by Joe Ackerman and Jeff Neal and John Sahi, for the Xenopus oocyte, and it's 4 orders of magnitude larger, 100 seconds. That's a huge range for this lifetime. And this is the result of many studies by many people. And the simple equation that I referred to, and you should remember, is that you can write, if you have a well-mixed compartment, and all cells are absolutely well mixed. Even the oocyte is on the verge of being well mixed, and Joe showed that very clearly. The mean lifetime is given by 2 factors. One of the factors is a permeability coefficient, and we're going to change PD to PW because you'll see why in a minute, but it means the permeability coefficient for water, the equilibrium water permeability coefficient. And some factor, claustrophobic ratio, that has to do with the size of the cell. And a lot of times I'm going to show you reciprocal T1, which is a rate constant for the equilibrium rate constant for unidirectional efflux. And it's still 2 factors. It's a permeability coefficient on the top and A over V. And that's how you interpret, that's how you can, if you know the size of the cell, you can go from the permeability coefficient to the lifetime. You can go, if you calculate how many water molecules, you can calculate the flux. In fact, 1 over tau I is that rate constant. So if you think about the red cells, it means that per second, on the average, the entire contents of the red cell is exchanged 100 times per second. And on the oocyte, it takes 100 seconds for the entire contents to get exchanged. Well, the transport physiologists who were studying this were doing all these experiments of inducing osmotic gradients and looking at unidirectional fluxes, and they call it the flow permeability, PF. And here's a famous member of the transport physiology community, Alan Verkman in San Francisco, And he kind of declared about 10 or 11 years ago, because of its limited utility, PD will not be discussed further. They've kind of gone away from the equilibrium water exchange, even though they started it in the 40s, and they've studied this unidirectional flux. Well, a few years ago, we decided to get into this game, and just as Bob Schulman mentioned, many experiments in his lab over the years started with yeast cells, and the same with our lab for 30 years. We've been using yeast cells. Gil Navon did a lot of studies with yeast cells at Bell Labs. And so we decided to, this is a modern picture of yeast cells, but we're going to put a paramagnetic agent on the outside, mostly it's Magnavis, gadolinium DTPA, and we're going to study the water exchange across the cell membrane of the yeast cells. And in fact, this is a modern version of, but what we do is a simple inversion recovery and measure the amplitudes. And if you make the right function, the approach to equilibrium, if it was a single exponential recovery, the log of that function versus time would be a straight line. And so when you have 9.3 millimolar paramagnetic on the outside, it's definitely not a straight line. And there are two components, and you could do an inverse Laplace transform of this recovery, and you can see that it's not monoexponential. As we talked about on Tuesday, you can't trust these very quantitatively, but they're fantastic for visualizing what's going on. It's very important to look at these, but it's really better to analyze the raw data with whatever exchange model you're going to do, and when the results I show you today are going to be based on analyzing that. But about 16 years ago, when we first did this, we actually looked at the stack plot of the relaxograms. This is T1 versus the concentration of gadolinium DTPA, and we could see that if there were no gadolinium DTPA on the outside, you have a monoexponential recovery with a single T1. If you titrate really carefully with gadolinium DTPA, at some concentration you see 2, and we could quite well assign this to represent extracellular water, O for outside, and intracellular water. So what's going on here, that is, this recovery is not single exponential. We're not changing the temperature. This is an isothermal experiment. The equilibrium exchange of water is constant. We're changing the shutter speed, which is the absolute difference of the 1 over T1s, such that here the shutter speed is very small and the exchange looks fast. Here the shutter speed is larger and the exchange looks slow. But it's not changing. It's the way the exchange condition is in the MR. This is a complicated plot, but it's 1 over T1 versus concentration. It's not linear. And you can fit the positions of these and the intensities. And here 16 years ago, we learned the average lifetime of water inside the yeast cell was 670 milliseconds. And with this suspension, 64% of the water was on the outside. Well, Jim Balshai, shown here at Fenway Park, decided a few years ago to really do this right, much better than the way we did it 16 years ago. And we hired Yaji Zhang. And they have done fantastic and really discovered something really very interesting. The busy slide, I'm not going to go through this is just that log plot at different concentrations of Magnavis to say that when there's none, this is really the raw data, it's a straight line, it's monoexponential recovery. When you have enough agent on the outside, it's not monoexponential recovery. But I want to point out, I won't go into details, it's not in the slow exchange limit. It's in the slow exchange regime. It's out of the fast exchange limit, but it's not yet in the slow exchange limit. We could go through that in great details. They showed they could make a stack plot of relaxograms, but they decided properly to really analyze the raw data, and for this particular case with 9.3 millimolar gadolinium, they used a much less, less cell density. Only 11% of the water is on the inside, and the lifetime was 540 milliseconds. So now the fun began. Jim is very observant and very careful, and one day he was looking at these raw data, and he noticed this is the plot of different concentrations when Camille, we learned how to bubble these cells very carefully. These were bubbled with nitrogen. They were anaerobic. And these cells were bubbled with oxygen. They were aerobic. And look, what happens to the recovery from inversion on the semi-log plot? When you go from anaerobic to aerobic, they tend to get straighter and with greater slopes. The slope is minus 1 over T1. Why on earth would the recovery from inversion care about whether the cells were aerobic or anaerobic? What's up with that? It suggests that the lifetime, so here's at several different concentrations, when you bubble with nitrogen, in this particular, and there are many studies for each point, many samples, when you bubble with nitrogen, the average lifetime is 560 milliseconds, kind of similar to what we got 16 years ago. But if you bubble with oxygen, the lifetime goes down to 295 milliseconds. The exchange gets faster when you bubble with oxygen. Well, what could be causing that? Well, most of you know a lot more about metabolism than I do. One of the main things that oxygen is doing, as we've heard from many people, is coming into the cell and being used to make ATP at the mitochondria. So perhaps, why on earth should the water exchange, equilibrium exchange, care about what is the level of ATP in the cell? So if that's what it is. So the first thing we have to do is see if that really is what's going on. Is it the change in ATP? One way to do that is to add dinitrophenol, which is well known to uncouple the production of ATP. So if you bubble with oxygen, but you add dinitrophenol to it, you get the same kind of lifetime you got if you were bubbling with nitrogen. So that suggests that this really is dependent on the level of ATP in the cell. And of course, with NMR, you can check this directly. And so Jim measured the level of ATP with NMR and also with liquid chromatography. And when you're bubbling with oxygen, you got a lot of ATP. And as soon as you switch to nitrogen, the ATP goes down, and it can actually go to almost zero. So then Jim and Yaji did a series of experiments. They used 3 different strains of yeasts. One of the strains was one that needs adenine in the growth medium so they could control the steady state level of ATP, and they could make them anaerobic and aerobic. What the result of it is that they could vary the ATP concentration over the steady state ATP over almost 2 orders of magnitude. And more or less, this is 1 over tau I, or KIO, the rate constant for efflux, unidirectional, is linearly dependent on the ATP level. If you go to the intercept where there's really no ATP, it's 830 milliseconds, and that gives you a permeability coefficient of 9 times 10 to the minus 5th. And I'll show you that that legitimately could be called PD, because here's a table from Finkelstein's famous monograph showing water permeability across model bilayer membranes. No proteins. The transport must be diffusive and passive. And if you have fluid membranes at 25 degrees C, which is the same temperature that we did the experiments, here's the value we get for the yeast cell. It's in between values for model membranes that don't have any proteins in them at 25 degrees C. So when you have no ATP present in the cells, it really is simple diffusion of water across the bilayer membrane. It's still quite fast, and I'll show you how fast in a minute, but it's simple diffusion. So what's up with the ATP dependence? When we get out to here, we've more than doubled the rate constant for water efflux, and we've gotten to 10 to the 12th water molecules per second per cell. The permeability coefficient has gone way up with ATP. All right, so we've established that the permeability coefficient for this is dependent on the level of ATP. What's the mechanism? How can it be doing that? Okay, you just finished eating lunch. You know that much of the ATP that you make from your lunch is going to be used to run pumps. In the case of the yeast cell, the major driving ATPase is a proton ATPase, not the one that's in the mitochondrion, but in the yeast cell membrane. It's driving protons out, but there's a sodium proton exchanger somewhere around here and a potassium transporter somewhere around here that acts as an effective sodium-potassium pump. And Jim reasoned that it must have something to do with the activity of this proton ATPase. And he looked in the literature and he found an herbicide that people had studied that turned out to be a specific inhibitor of the proton ATPase of the yeast cell. It's an organoselenium compound of all things. It's called epsilon, and when he switches from nitrogen to oxygen with no epsilon, here's the huge increase in the water exchange. If he adds epsilon, it's mostly inhibited. And in fact, he showed that that did not inhibit the production of ATP. In fact, you even get a little bit more ATP with epsilon present than without. But it's not inhibiting the ATP. It must be the activity of the proton ATPase itself is implicated in the water exchange. Now, I want to point out that when you go from nitrogen to oxygen, there is a small shrinking of the cells. This is the mole fraction of water on the inside. But it's very transient. It only lasts about 15 minutes. It's very small. It's a 5% reduction in the radius, and it's very slow. It's 3 orders of magnitude slower than the equilibrium rate constant. So you can have net fluxes that might go on parallel, but the equilibrium exchange is 1,000 times faster, and it's dependent on ATP. And if you get a mutant of yeast that are heterozygous for the proton ATPase, that means they're missing half of them, it turns out that, in fact, the water exchange is the same as if you didn't have any oxygen present. So we really have implicated the activity of the proton ATPase. Now, we still need a molecular mechanism of how is it that this thing, which is transporting protons, is causing water transport. Well, at the end of last year, a wonderful review article came by Thomas Zeuthen in Copenhagen that, in fact, in the last few years, people have discovered that many membrane co-transporters of, say, potassium and chloride or sodium and potassium chloride also co-transport water. And the stoichiometry is big time. 500 water molecules for every chloride and potassium, or 590 for each for those. There's a lot of obligatory water co-transport going on parallel to the co-transport of solutes. And I don't have time to really show you the data, but it's really beautiful data, and it actually means that you can make water go against an osmotic gradient if it's using energy of the solutes going down their electrochemical potential. And that's really important in your intestine. If you think about it, you have to be able to get water out against a gradient. Well, Zeuthen speculated on what the mechanisms might be. Here's one that I thought of at first. If solutes come through, they might make a local hyperosmolar a patch on this side of the membrane and then suck water through somehow because it's hyperosmolar. Or you might have a hyperosmolar cavity that would suck water through. Well, those are interesting, but they're really not the most elegant. Here's a cartoon by Stan Schultz saying, well, maybe you have some water transport proteins that happen to be right next to these, and the solute goes through and it sucks water through. But in fact, Zeuthen looked at the literature, and in the last few years, a number of proteins have been found that don't just have narrow channels, but actually have water cavities. Here's a potassium channel with a cavity. Here's a chloride ion transporter with cavities. There are some that do not. Here's a calcium ATPase. Here's another compound. There are a lot of structures. And here's a proton ATPase from the yeast cell, and here's a water cavity. There are kind of large cavities. There's some others with these two, and I have to flash forward. And if you have some some conformational changes of the protein subunits, you can expose these cavities to one side or the other of the membrane. And so he drew the cartoon of this wonderful mechanism that I like to think of as a canal lock mechanism. And here comes the solute, which is the little black ball, into the lock of the canal, and there's a specific binding site for it, but a patch of water comes with it. And then you close the gate behind the boat, and then you open it on the other side, and every time a solute goes through, a large stoichiometric amount of water goes through. And so here is a cartoon to help us just think about this. So here's a yeast cell cartoon. If you had no ATP present, then it's the simple diffusion of water across the bilayer membrane. And that's fast. It's 10 to the 12th water molecules per second. But if you increase the ATP to a normal kind of level, you crank up this obligatory water cycle that can be 2 or 3 times as fast as this one. Now, we don't know whether the canal locks are in the proton ATPase or in the potassium channel, as I showed. These are just representative. There are many transporters in the cell membrane, but integrated over the whole cell, you know that it's cycling solutes, and I won't have time to show you, but we have evidence in the cardiac cells in intact heart with the sodium potassium pump. You know you're cycling solutes all the time because you're pumping sodium out of the cells and it's leaking back in, so you're cycling it. Who knew that at the same time you're cycling water big time? And you could say, why did nature evolve this system? We could flatter ourselves to say it evolved it just so we could discover it by NMR, because NMR can measure this. But I really don't think nature cared too much about NMR. My pet theory is that it's like when you have to turn your car on and have your RPM of your engine idling before you can engage the gear. You have this giant water cycling going on that's really 1,000 times faster than unidirectional transport. And if an osmotic gradient arises, it takes care of it instantly. And it can handle large changes in osmolytes. And actually, I think this has implications for many of the metabolic features that have been mentioned in the last 2 or 3 days. And we started measuring tau I a long time ago in DCE MRI, and this offers a metabolic dimension to DCE MRI. So before Mikhail pulls me off the stage, thank you very much.
Thank you very much.
Thank you. Camille.
You are focusing on the water exchange, but when I look at your diagram right there, those channels are exchanging hydrogen ions, and of course you are measuring proton D1. So why couldn't just be the hydrogen ions exchanging be responsible for this process that you're seeing?
It's a great question, but they're not enough. The protons that are the difference in protons between the two sides are used to create the membrane potential. And as you know, that's an exponential, so it's very, very tiny. I mean, pH is near 10 to the concentration near 10 to the minus 7th on each side. So in fact, it actually is water molecules, because in the heart, which we have data but I haven't shown you, it's not pumping protons, it's pumping sodium, and we see the same thing.
Just just a follow-up question. So in principle, of course, there are not enough protons, and then the net flux of the ions, the rate at which they go, can be relatively slow. But you could have a exchange that is going back and forth relatively fast, as opposed to the net.
It is. Sodium, in most cells, sodium is going back and forth 10 to the 8th molecules per second per cell. And we have some evidence for that. And water is just going with an increased stoichiometry. But you're cycling sodium across your cell membranes very fast right now. But there's no net change in sodium unless you do something really drastic metabolically.
So you don't think then this net, not net, but this going back and forth without a net change, and then at the same time shuttling the protons back and forth cannot account for it.
Well, possibly it could in the yeast, but I really don't think so, and it certainly wouldn't in the heart. So,
Continue.
good question. Yes.